You and the organizing committee for inviting me to such an exciting conference in such a beautiful place. The only problem is when I left Canada it was minus 31 degrees Celsius, and when I got to Turkey it was plus 18 degrees Celsius, and Canadians aren't very good at adapting to warm weather at this time of the year, so I've picked up a bit of a cold, but I think my voice will hang out. So I'm going to do a brief overview on advances in antidepressant targets and how that's may develop or may influence drug development. We've had already had some symposia in this conference about glutamate and depression, for example, where we've covered a lot of this information, so some of it I'm going to go over quite quickly. The currently available prescription antidepressant drugs, as we know, affect reuptake, metabolism, and/or receptor activity of the biogenic amine transmitters, serotonin or 5-HT, and/or noradrenaline or norepinephrine. These drugs are associated with response and remission rates that are lower than we would like. They produce excessive adverse effects, and there are prolonged periods before clinical improvement actually occurs. So these are, we'd certainly like to have better drugs than we currently have available. So I'm going to discuss today some new possible targets. We've had in the last 15 years or so some elegant neuroimaging techniques, and these have improved considerably as we've got stronger magnets. We've had very sophisticated molecular biological and neurochemical and pharmacological techniques available, and these have helped us identify several exciting new possible targets for the development of future novel antidepressants. So the biogenic amine or monoamine hypothesis of depression was put forward about 50 years ago now, and taken in its various forms. This is a summary. Depression is a result of a functional deficiency of noradrenaline and/or 5-hydroxytryptamine at specific synapses in the central nervous system. So this has been a very useful hypothesis for developing drugs. But we've known right from the start that it's oversimplified. When you're working with the brain, you can't just deal with one or two neurotransmitters usually. And it was pretty obvious early on that other neurotransmitters and neuromodulators are also involved. I'm going to discuss some of these today, including amino acids, GABA, and glutamate. We keep coming back to the interaction between those two amino acids. Peptides. I'm going to mention briefly corticotrophin-releasing hormone and substance P. Steroids, I'll talk about the neurosteroids a little bit. Melatonin and neurotrophic factors. And all of these have in common their involvement with stress and with the HPA axis. So, some other possible antidepressant targets. As Serdar mentioned, dopamine's been the focus of attention for many years in schizophrenia, but has been largely ignored in depression, but may well be important. It's involved in reward mechanisms, it's involved in locomotor activity, and could well play an important role in depression. A number of companies right now are looking at developing what we call triple reuptake inhibitors. These are drugs that inhibit reuptake of serotonin, noradrenaline, and dopamine simultaneously. I can see some problems here when you start to develop drugs where you've got three properties built in. You may get drugs where you, for each individual patient, you don't have the optimum ratio of noradrenaline to serotonin to dopamine reuptake, and this could cause problems. So it'll be interesting to see how these drugs make out when they actually undergo clinical trials. Amino acids, we've seen at various symposia at this conference the importance of the balance between GABA, which is a major inhibitory neurotransmitter, and glutamate, which is a major excitatory amino acid neurotransmitter. And the delicate balance between these two seems to be upset in a number of neuropsychiatric disorders, including depression. So with GABA, there's been evidence for quite some time that plasma GABA levels are low in major depressive disorder. And with the development of sophisticated magnetic resonance spectroscopy techniques, we've seen that brain GABA levels have been reported to be low in some brain areas in depression. And even drugs like the SSRIs, where we think of it being relatively specific for serotonin, have been reported to result in an increase in brain GABA levels. The MAO inhibitor antidepressant phenelzine, which has been around for a very long time, actually also inhibits GABA transaminase. Most people don't think of that, and causes a marked increase in brain levels of GABA. After chronic administration, antidepressants of several types can alter the ratios of the isoforms of the GABA-A receptor subunits, and this happens after chronic and not acute administration. And this has been shown with phenelzine and some of the tricyclic antidepressants. Numerous studies in body fluid samples from depressives and preclinical drug studies suggest that there's hyperglutamaturgia in depression. However, many of the MRS studies don't support elevated brain levels of glutamate in depression, which is a bit of a problem right now. However, reports on rapid response to ketamine that's been discussed at several symposia here this week seem to support the concept of excessive glutamatergic activity in depression. So although rapid clinical antidepressant response still remains relatively elusive, as many of you know, recent studies with the NMDA glutamate receptor antagonist ketamine are promising, and I won't go into much detail on this because it's been covered very nicely in a number of other symposia this week. And as we all know, ketamine can have a number of adverse effects as well, so it's certainly not an ideal drug at this time, but very exciting that it is causing the rapid clinical response. So combinations of ketamine with other drugs to counteract side effects are under investigation. Riluzole and trexopradil, which are also NMDA receptor antagonists, have also shown promising results in preliminary studies, as has memantine as well. So studies on glutamate in depression suggest strongly that actually multiple receptors are involved. It's not just NMDA receptors. So we have the NMDA receptor, AMPA receptors, and various metabotropic receptors. And this makes interpretation of the mechanism of action of ketamine difficult, but raises the possibility of discovering various interesting new antidepressant drug targets. So advantages and disadvantages there. Serdar brought up glial cells, and indeed these are important. There's 10 times more glial cells in the brain than there are neurons. And originally we thought these cells were just there for supportive purposes for the neurons. But in fact, we now know that they can have a major role in neurotransmission as well. Glial cells are involved in uptake and release of glutamate, and there may well be a dysfunction of glia-neuron interactions in depression. Serdar also mentioned, when he was talking about schizophrenia, glycine and D-serine. These are potent coagonists at the NMDA receptor, and it'll be interesting to see if they're also affected in depression. There hasn't been much work done in this area. More of it's been done in schizophrenia. Neuroactive steroids is another area that Serdar mentioned with regard to schizophrenia, but these have also been studied quite extensively in mood and anxiety disorders. These are hormones that exert rapid non-genomic effects at ligand-gated ion channels. most of the work's been done with the GABA-A receptor and the NMDA receptor. Examples are pregnenolone, allopregnanolone, dehydroepiandrosterone, or DHEA, and then the sulfated forms of pregnenolone and DHEA. Just here to show you some structures, the typical sort of steroid structure with the 4 fused rings. So these can act at receptors for several neurotransmitters, but most of the research on them has been done with regard to the GABA-A receptor and the NMDA glutamate receptor. Some of them act as positive and some act as negative allosteric modulators at these receptors, and I'll just show you some examples here in a minute. And the balance between these neuroactive steroids is thought to be important in the etiology and pharmacotherapy of several mood and anxiety disorders. They've been studied relatively extensively now in, for example, panic disorder, social anxiety disorder, and they seem to certainly levels are altered in those disorders and seem to come back to or to normalize after successful drug treatment. The SSRI antidepressant fluoxetine is very potent at elevating brain levels of allopregnanolone. In fact, it's more potent or has a stronger effect on elevating allopregnanolone than it does at inhibiting serotonin reuptake. So this effect actually occurs before you start to see significant serotonin reuptake inhibition. And allopregnanolone is interesting because it's a neuroactive steroid that is a very potent positive modulator at the GABA-A receptor and appears to have antidepressant, anxiolytic, and neuroprotective properties. A lot of this work was done in Mimo Costa's laboratories with Dr. Pinette. This is just a slide showing you the synthesis of the neuroactive steroids, and they can be synthesized in the brain, in which case they're called neurosteroids. And you can see here pregnenolone has a very central role to play. It can be synthesized by various enzymatic pathways to give allopregnanolone or TDOC or DHEA down here. And so we've got a number of pathways here that we can act on with various drugs to see if we can alter levels of these neuroactive steroids. And we just published a paper in Brain not too long ago showing where we did a gene array and showed that a number of these enzymes were disrupted or were inhibited in multiple sclerosis. And when we did the analysis of the neuroactive steroid levels at the same time in postmortem brain from MS patients and found a very nice correlation. So it's very exciting now to see what happens in depression looking at these neuroactive steroids. So allopregnanolone and TDOC are potent positive modulators at the GABA-A receptor, so they have very strong anxiolytic effects. DHEA is a positive modulator at the NMDA receptor, and the sulfated forms of DHEA and pregnenolone are negative modulators at the GABA-A receptor and positive modulators at the NMDA receptor. So you can see that the ratio amongst these various neuroactive steroids may be very important in determining symptoms in a number of neuropsychiatric disorders. So peptides, I'm going to discuss 3: corticotrophin releasing hormone, substance P, and glucocorticoids. The HPA axis, as I said, is affected by stress. It's affected by most of the neurotransmitters that we're interested in depression, including serotonin, noradrenaline, GABA, glutamate, and the neuroactive steroids. As you know, the HPA axis is hyperactive in many depressed patients, and this is indicated by increased secretion of corticotrophin releasing factor or hormone, CRF or CRH, adrenocorticotropic hormone, ACTH, and cortisol. So CRF or CRH is a major component of the HPA axis, and Dr. Nemiroff and his colleagues have done a lot of work in this area. We know that levels in CSF and plasma have been reported to be elevated in depressed patients, and remission seems to be associated with normalization of those levels. Preclinical studies suggest strongly that excess CRF is associated with production of symptoms of depression, so these are studies in lab animals, and certainly CRF antagonists are being investigated by a number of companies as potential antidepressants. Substance P, there have been some promising reports on substance P receptor antagonists. These act at neurokinin receptors, and suggestions that they may be effective antidepressant and anti-anxiety agents. Pierre Blier from University of Ottawa has done a lot of work in this area, and his studies suggest that these NK1 antagonists produce effects on serotonin and noradrenaline that are quite similar to those produced by other antidepressants. The glucocorticoid receptor 2, or GR2, activity is increased in mood disorders, and a drug called mifepristone is a GR antagonist and is in phase 3 development. There's some promising results in psychotic depression and in bipolar depression with this compound. It's interesting, it rapidly increases adult neurogenesis and also blocks corticosterone-induced 5-HT2A receptor upregulation. Melatonin, again, there was a symposium on this at this conference on agomelatine. Melatonin, as you know, is involved in regulation of sleep, and sleep disorders are very common in depressed patients. Agomelatine is an agonist at MT1 and MT2 melatonin receptors, and an antagonist at the serotonin 5-HT2C receptor, and has been reported to have good antidepressant properties with quite a favorable side effect profile. I think most of you know too that some of the atypical or second generation antipsychotics and their metabolites appear to have quite good antidepressant properties. This has been studied with, for example, olanzapine, aripiprazole, and quetiapine. In the latter case, the N-desalkyl metabolite has the antidepressant properties. Deep brain stimulation, some exciting work in this area over the last 5 or 6 years. This has been used successfully for some cases of treatment-resistant depression. This involves chronic stimulation of white matter tracts adjacent to Brodmann Area 25, and this is from Mayberg Lab. Sid Kennedy in Toronto has also been involved with this as well. The immune system in depression, not only does it seem to be important in schizophrenia, but also in major depressive disorder. And again, we come back to glial cells, the microglia that can release cytokines. Cytokine theory of depression proposes that alterations in the immune response mediated by these cytokines can result in some of the behavioral, cognitive, and neuroendocrine changes that we see in major depressive disorder. The cytokines that are released by the microglia can be pro or anti-inflammatory, and obviously the ratio between those is important. And it's been postulated that depression may be caused by, or at least related to, excessive amounts of pro-inflammatory cytokines such as interleukin-1 and tumor necrosis factor alpha and TNF-alpha. It's interesting here that pro-inflammatory cytokines are actually stimulators of the HPA axis, so that fits in with hyperactivity of the HPA axis, and can decrease 5-HT levels by diverting metabolism of tryptophan away from the formation of 5-HT, so you would get a functional deficiency of 5-HT as well. So depression may be related to excessive amounts of pro-inflammatory cytokines, and more studies in this area will help clarify the role of glial cells in depression, but this is certainly becoming a very exciting area now, looking at glial cells, the role in neuropsychiatric disorders, and how glial cells and neurons interact with each other. Administration of interleukin-6 and TNF-alpha to rodents can produce anatonia, psychomotor retardation, and reduce sexual activity. Those are things we see in depression. Cytokine levels have been reported to be increased in major depressive disorder, and reduced in response to antidepressants in some but not all studies. Just a diagram similar to the one that Serdar showed you, showing that not only do we have neurons, but we've got the glial cells, so the microglia, the astrocytes, the oligodendrocytes, and certainly with the astrocytes and microglia, they can be involved in uptake and release of things like glutamate. So I'm just going to finish off with a few slides here talking about other possible antidepressant targets, including signaling cascades, neurotrophic factors, neurogenesis, cyclic AMP response element binding protein, or CREB, and BDNF, or brain-derived neurotrophic factor. And this is a quote from a paper by Trevor Young about 9 years ago now. As the neurobiology of mood disorders and the mechanism of action of antidepressant drugs continue to be elucidated, there's been a shift in emphasis from changes in neurotransmitter release and reuptake and metabolism to regulation of gene expression and neuroprotection. Evidence from animal studies suggests that antidepressant drugs may act on specific transcription factors and target genes that regulate processes such as neuroprotection, and certainly has been an emphasis on this aspect of antidepressants for the last few years, the last decade. So this is another hypothesis. The neurogenesis hypothesis of depression states that depression is a consequence of impaired neurogenesis in the hippocampus, and that antidepressants exert their effect by stimulating that neurogenesis. Clinical studies have provided evidence that recurrent depressive episodes are associated with reduced hippocampal volume, and preclinical studies, and some studies now coming out clinically as well, have provided evidence indicating that chronic antidepressant treatment results in an increase in hippocampal neurogenesis, and that the behavioral effects of antidepressants do not occur when you block that or prevent that neurogenesis. Brain-derived neurotrophic factor, or BDNF, has received considerable attention as a target for antidepressant drugs. In animal studies, chronic and repeated stress has been shown to decrease levels of the messenger RNA for BDNF in the hippocampus. Chronic administration of antidepressant drugs from several classes, so tranylcypromine, an MAO inhibitor, sertraline, an SSRI, dizipramine, a tricyclic antidepressant, phenelzine, an MAO inhibitor, and myanserin, sort of an atypical type antidepressant, significantly increases BDNF messenger RNA in the rat hippocampus. And this occurs after chronic but not acute administration, so it fits in with the clinical delay. Also, preclinical studies suggest that the antidepressant effects of ketamine may be related to the rapid synthesis of BDNF. We also know from studies in the forced swim test for antidepressants that the behavioral despair that you see there can be alleviated by midbrain infusion of BDNF. Serum levels of BDNF have been reported to be lower in drug-naive depressed patients than in antidepressant-treated depressives or in healthy controls. Serum BDNF levels have been reported to increase to normal after successful antidepressant treatment. In studies in vitro, phenelzine, which is an MAO inhibitor antidepressant, increases levels of messenger RNA for BDNF in both astrocytes and neurons. And but some Duman's lab, and this is where a lot of the BDNF work's been done with antidepressants, has said that deletions or antagonism of BDNF does not always produce depressive-like behavior. And Rantamaki, in his paper, said that it's not clear at this time the extent to which the different antidepressants differ in their ability to alter BDNF levels. So we still need some comparative work done there on antidepressants. So chronic depression may represent a subtle neurodegenerative disorder, and early and vigorous treatment of acute episodes may be important in the prevention of enduring dysfunction. So we've got in many places in North America, and I assume Europe and Asia as well, there are early intervention psychosis clinics for schizophrenia, but there's now predictions that these may start to increase for depression as well as schizophrenia. There are certainly ever-increasing reports in the literature on the effects of antidepressants on neuroprotective and anti-apoptotic proteins. So neuroprotection and antidepressants is a very active area of research at the present time. So, in summary, there's an ongoing search for novel antidepressants that are faster acting, have fewer side effects than those that are currently available. Research in this area in the near future will probably expand in the following areas: excitatory amino acid receptors. As I said, with ketamine, we still don't have a really good handle on the mechanism of action there. Neuroactive steroids, neurotrophic factors, neuropeptides, and very important, I think, the effects of antidepressants on both glia and neurons and the interactions between the two. Increasing availability of stronger magnets with which to conduct magnetic resonance spectroscopy is also going to allow more complex studies of glutamate and other amino acids in the brain, for example. We can look now more closely at hippocampal subregions. So acknowledgements, these are colleagues I worked with. Catherine Todd is the interim chair of our department. Gail Rao and George Van Newton, who've been technical assistants of mine for many years, and Sarah Tomlinson, our administrative research officer in the department. And I'm just listing a review here, a paper here by Sid Kennedy, Alan Young, and Pierre Blier, which I think is very worthwhile reading in Journal of Affective Disorders. It's called Strategies to Achieve Clinical Effectiveness, Refining Existing Therapies and Pursuing Emerging Targets. Thank you.